Toc. Non, c'est pas ça. Non, ça c'est. On va faire juste. Donc, okay, so Xiaoyin, that's a pleasure to see you again, and I will just share your file. Okay, so I don't know what happens if we see the right things on the blackboard, but it should be. I think you can speak. We record. I think it should work. I will maybe connect and my telephone to stop. Thank you. Tu veux pas, je sais pas comment la voir elle. Il faudrait mettre.
C'est pas grave.
Je vais essayer d'arranger ça avec mon téléphone.
Okay. So thank you for inviting me. And today I will be talking using statistical and computational methods to identify genetic variants in large-scale genomic data. So I am Xiaoyin Li, a former PhD student of Portugal. I finished my thesis in 2014, and then I left France, went to the United States. I did 4 years of postdoc at Case Western Reserve University, and then I found a faculty position at St. Cloud State University. Yeah. My research focuses on using statistical and computational methods to deal with high-dimensional, large-scale genomic data. And I will start with, first I'll introduce the background, and then my research is mainly focusing on addressing 3 questions. So the first one is, how can we identify disease-related loci, disease-related genes? How can we identify those genes? The second question is, which genes in the genome govern the co-occurrence of disorders? That's a phenomenon, what we call pleiotropy, but I will introduce that later. And the last one is how we understand the mechanism underlying those genes that influence pairs of traits. Okay, so everyone heard about genes, but what are they? Genes are segments of DNA in everyone's cell, and some genes determine how we look like our hair color, eye color, skin color, and some genes are involved with the likelihood of develop some health conditions such as diabetes, hypertension. So each person has around 20,000 genes in your body. And so no, this is the first time. Okay. All right, so genes are made of DNA. So what are DNAs? DNA is made up of 4 building blocks called nucleotide. We use letters to represent them. G, C, T, A, those letters made of DNA. And in human body, DNA is found inside of this nucleus. Most of the DNA is found inside of this part, and some are outside of the nucleus, which we call mitochondria. But this is the DNA that we are mainly focusing on. And in this nucleus, the DNA is formed the chromosomes. It's a helix shape, and it has 2 strands. So the nucleotides attached to each other, which we call the base pair. So this is a base pair. A attached with T, G attached with C. So if we know one letter in this side, then we will know the other one automatically. So those are the letters, those are what we call base pairs. And there are many, many base pairs in human body. And so you and me, we share 99.8% of those base pairs. That means 99.8% of the base pairs are identical among humans. There are only 0.2% there are different. So the 0.2% variation determine who you are, determine why we look different. That's the 0.2% that we are interested in. Okay, so let's talk about the 0.2% variance. So there are mainly two types of variations we are talking about. The first one is what we call the nucleotide level, so the base pair level. You can see that. For example, here, those are identical.
And just one question. When you do 2 8, that means on the 2 frames.
No, just one screen, because it will only record the letters for one screen. Yeah, just one. Because if you know one, then you know the other one automatically. So you don't need to record the letter for the other one. So that's one.
Because in our all work together, we were allowing all the sequences, and I think that some sequences in the DNA are forbidden. We take it into account.
Yeah, that's because you have two chromosomes, is two chain, and each chain is a helix. So technically, for each position, you have 4 letters, but you just need to know one in each chain.
Okay.
Yeah. Okay. So those are called nucleotide level variation, because it's just a single base pair changes. So we can see that here we have some individual carry T allele and some individual carry A allele. And those are what we call the structure variation. That means just a sequence of DNA that changes. Usually it's being deleted, or it's insertion, or it's just the other way. So what we are interested in is this base pair. So very basic base pair changes. We are interested in this type of variation. So we can see that here we have 12 individuals on those genomes that they are identical. At this position, we see some variations. Those are the genetic variants that we are interested in, and we call it SNP. So SNP is called single nucleotide polymorphism. It's just a single changes in a nucleotide base. It occurs normally throughout a person's DNA, and it's very common. So it occurs almost once in every 1,000 nucleotides on average. So some of them just the changes, they are not associated with any disease, but some of them are associated with some phenotype. So that's the variation, the variants that we are interested in and we want to study. Okay, so this is a close look about a SNP. So we can see that this is just one person's genome. We have on each chromosome, you have 2 helix, and you have 2 chains. So you know this position, then you will know this one automatically. So we record this one only, and here for on the other chain, we record this one. Make sense?
So it means that SNP means these are the locations where these 0.2% that you were talking about. That's something that.
Yes, the SNPs are the variations that we are talking about. Yeah.
And the rest is the same for everybody.
Yeah, right. Rest are the same for everyone. Yeah. Okay. So how we read those SNPs? We use this SNP array. It can measure up to 100,000 SNPs, and it costs. So before it costs like $1,000 for one to genotype one individual, but now with the development of technology, it costs less, like $100 to genotype one individual. And the technique, the chip that we use, this is what we call the SNP chip. So there's 12 tips, so it can genotype up to 12 individuals at 10 to 6 SNPs. Okay, so this is a picture of my my genetics results. So a year ago, I was curious. I know that I'm from Asia, but I'm just curious about my ancestry. And we can use this genotype. We can just genotype you and determine where you're from, your ancestry. So this is my results. I have 99.11% Eastern Asian ancestry and 0.89% European. Yeah. So if you've seen my son, you understand why his hair color is light. Yeah. So that's very interesting. And this is just one of the things that people make this genetic thing commercial. So in United States, it's very common. It's called 23andMe or Ancestry Kit. You can buy that at any pharmacy, and you just do a swap and send them back. Yeah, they should be. So actually, yeah, they should tell me which part I am from, but I guess it's this is just an overall results. Yeah. So in some companies, they'll give you more precise results. This is just a free one, so they just give me an overall results. But if you buy those kits, then you do a swap, send it back to the lab. They'll send you the result, tell you like how much percent you are from Finland, how much percent you are from France, German or Italy. Yeah.
So that's from 23andMe.
This is from Ancestry, another company. But the biggest company is 23andMe. They are dominating in the market. This is another company. Yeah.
I also tell you you might get diabetes or whatever.
Right. Yeah, it's another test. So it's not the same one. Yeah, maybe you have to pay more just to get to see if you are carrying some risk allele that will increase your risk of develop some health conditions. Yeah.
It's not Germany, it's Germany.
Okay, thank you. My teacher. Okay. All right. So okay, so how can we identify disease-related gene loci? So what we do is we call this genome-wide association study. And this is just a rapid scan of your genome. So we genotype you for each individual. We have like around 100,000 SNPs, the A, G, C, T, those letters for each individual. And then we just do an association from the genotype to phenotype. So we are looking for associations, which genetic marker is associated with a certain disease. So it's just a simple regression model. Depending on the phenotype, if we have quantitative phenotype, then it's just a general linear regression. And if we have binary traits like disease or not, type 1 diabetes or healthy individual, then it's a logistic regression. It's just a regression model. So the thing is just a rapid scan because we have around 100,000 SNPs. So we need to do the regression for each SNP at a time. So it's 100,000 tests. So what we are aiming is to identify which region or gene or SNPs in the genome are associated with disease or certain phenotype. And the study design is we take into consideration the population structure. We know that, for example, the African Americans, they are likely to develop hypertension. So their prevalence of getting hypertension is higher than the European ancestry or Asian ancestry. And for Asian ancestry, for Asian people, Asian population, they are more likely to get type 1 or type 2 diabetes. So we need to take into consideration the population structure And if we are having quantitative phenotype, we use general linear regression. If it's binary traits, we have logistic regression. And then we genotype 1 million SNPs for each subject and determine which SNP is associated. Okay, so this is the study that I participated 5 years ago, and we got it published last year in Nature. So what we did is we have 5.4 million individuals of diverse ancestries. So we have Asian population, African American, European, Hispanic, etc. And in total, we have 281 studies. And we are looking at association between SNPs and height, human height. We want to understand how the genetics contribute to human height.
So association means correlation, or is that something?
Yes, correlation, yeah. Okay, so let's break this down a little bit further. So what we did is just a simple, a general linear regression. So for the regression, for the explanatory variable, we have only three possibles. 0 means you did not carry 0. You did not carry any of this allele, defect allele. 1 means you have 1, and 2 means you have 2 effect allele. So this is an example that there's no association because we cannot see a regression line. This is well, the line is flat. That means there's no association. Negative. Yeah, it can be negative. Yes. Yeah. This is just that example. Yeah, it can be negative. Yeah, you're right. And here, this is an example of we have a SNP that is associated with the height phenotype. So we can see that when the effect allele when the number of effect allele increases, then the effect size, the y-axis increases as well. So that's just it's a very basic model. There's very simple model, but we did this for each of the SNPs, for 1 million SNPs. And we are testing if any SNPs that are associated with the phenotype height. And because we did so many tests, 1 million tests, so we need to control for multiple comparison. So the p-value that we set to be considered, so in order to be considered a significant association, the p-value must be less than 5 times 10 to negative 8. So 5 times 10 to negative 8. That's the significant level for us because that's the independent SNP that we consider in the genome.
So what is the so each SNP has that's one locus in your genome, right?
Yes.
So that means there is one allele at this place in this SNP.
So can you say it again?
Yeah, I was wondering what if you have a look at one SNP, it means that you have a look at one very specific
Yes.
So how can you have 2 alleles?
Right. Let me go back here. Okay. So this is an SNP. Okay, so for this person, it has G allele and A allele. But for some other individual, they have G and G, and for some other people, they have A and A. So yeah. So because you have two chains, one chain is a helix, and you have 2 chains. So you have two letters. Makes sense. Yeah. Sure. Yeah. Yeah. When I started this, I confused this with this as well, but yeah.
We have 2 different genes. That means on the light is the graphic.
Yeah, here that's
It's
2 genes. Yes, we record that. Yeah. Good.
Okay.
Okay.
Thank you.
Yeah. Okay. Where are we? Here. Okay. All right. So after we did this association in each of the study, then we meta-analysis that because we did this individually, because some study, for example, I am the principal investigator of my study, which is called Cleveland Family Study. We only have around 700 individuals. We have the genotype, so I am responsible for that study. And people from all around the world, they have their study, so they did the same thing. And then once we have the result, we meta-analysis that. Meta-analysis will increase the power. So after we meta-analysis this 281 study, which include 5.4 million individuals, what we got is, this is just this is just a map to show where the study from. So we have individuals from all over the world. We have a lot from European, some from American. I think those red one and those yellow one are my study. So Cleveland Family Study, we have 700 European ancestry, and we have 700 African American. And then we have some from Hispanic, some from Asian as well. So those are our population. And what we found is, this is the result. We found over 12,000 genome-wide significant SNPs that are associated with height. So this much, 12,000 SNPs, 12,000 SNPs. That associated.
Means that the covariance is
0.01 or 0.1. Yeah. So 12,000 individual and almost has a independent SNPs that are associated with height. So this is the largest GWAS study for human height in the history.
But did these different studies have the same assumptions and the same setup, or can
Just the same assumptions, that same setup, yeah, because this is so the data is there. Usually for this type of study, we recruit individuals and then we genotype them. So their genotype, because it's from the blood, so the genotype does not change, so it will stay there. And then the phenotype, we also record the phenotype. And then this is just massive data. You can do a lot of things with that. So my research focuses on those data, but I did a lot of things, different things with those data. Yeah, this is just one study. We have a study pipeline, so we follow that. That's from the Giant Consortium. I'm part of the consortium. I'm a member of the consortium, and we follow the pipeline. So we did everything pretty much the same. Yeah, yeah, just to have a consistent results. Yeah.
So this was already done, having this meta-analysis in mind.
Yeah, this is already done. This is published last year in Nature. Yeah. And there is another study. So it's another phenotype, but basically the same thing. We have the genotype data and we have the phenotype data. So we looked at the association between SNPs and lipids. So HDL, high cholesterol level, low cholesterol level, and TG. So just the different measurements for lipids. And then this is also published in Nature two years ago, 2021. Yeah. So we have 1.65 million individuals, and how many SNPs we found? We found 337 I'm sorry, 773 lipid associated SNPs that regions that contain 1,765 variants that reach the genome-wide significance. So you can see that for different phenotypes, even though we pretty much use the same data, for different phenotypes, the SNPs are, the number of association are quite different, because height is a highly polygenic trait. It is like, so the heritability for height is like 50%. So you would expect more SNPs that are associated with height. But for lipids, this is well, more like a lifestyle disease. So the genetics contributed to lipids are less. So you would expect to see less SNPs, less variants that are associated with this phenotype. Okay. All right. So the next topic is pleiotropy. So what is pleiotropy? So pleiotropy is when a variant or a gene influences multiple phenotypes. For example, this one, this is a gene that is associated with, can see very well, with heart failure, with eye disease and the liver disease. So this is a well-known gene that is associated with 3 different phenotypes. So that contributed to 3 different phenotypes. So we are interested, because when we do the GWAS study, we found that many SNPs that are associated more than one trait. So some genes that are associated with height and other traits. So we want to know which SNPs are associated with more than one trait and why. So that's the next topic. So we are looking at which loci in the genome governs the co-occurrence of disorders, which loci, which SNP in the genome associated with more than one trait. So how we can detect them? So there are 2 types of studies that are proposed to identify those SNPs that are associated with more than one trait. The first one is called cross-phenotype association. When that means you have, so when you scan the genome, you take one variant at a time, but you have multiple phenotypes. Then by doing this, you will be able to find association between one variant and multiple phenotypes. And another one is multivariate regression. Well, the response variable will be a matrix. That's what I'm talking about. The multivariate regression. You have one variant, and then the response variable is multiple traits. Another method is what we call the univariate regression. So, we just looking at the regression separately and then combine the result together to see which one data point for each individual in the study. Okay. So the method I'm using is called cross-phenotype association analysis. And I'm using the summary statistic. It can integrate association evidence from multiple correlated, continuous, and binary traits via summary statistics. So when I talk about summary statistic, it's this results from linear regression results. So we can, this is very, this is the type of data I am working with right now for each SNP, because when we did the genome-wide GWAS study, we have this type of result. So this is the effect size, the B, the beta estimate, the standard error, the p-value, and the sample size. So for each SNP, we have this. And the reason I'm using this type of data is because this is publicly available. It's very easy to access. If you want to use the genotype data, a lot of time it has, if you don't own the data, it's just hard to get access to those data because it involves it's human genetic data. So a lot of time getting access to those genotype, raw genotype data is just hard. But this summary level data, this is publicly available. A lot of time, this is publicly available. So I have access to those data, and this is very good. So the method I use is called cross-phenotype association analysis. We use summary statistics instead of individual level data. And then we just meta-analysis those data. Then because we combine multiple phenotypes together, that will increase power. Okay. So this is an comparison of using cross-phenotype association data versus conventional GWAS. So we can see that our method can identify more genetic variants than the conventional GWAS study. And here is a picture of when we combine 3 traits together using the cross-phenotype association analysis. We combine height, BMI, and waist-to-hip ratio adjusted for BMI, because those 3 traits is what we call anthropometric traits. They are correlated. So if we combine those 3 traits together, that will increase power to detect variants that are associated with any one of those 3 traits.
What was it, BMI?
BMI, body mass index.
Yes.
Yeah.
The other two?
With the hip ratio,
Okay.
adjusted for BMI. After we adjust for BMI, the with hip ratio. Yeah. So those 3 traits, because they are anthropometric traits, just represent human body, height or the shape. Yeah. So they are correlated. So it makes sense to combine those together because they are correlated. If genes contribute to anthropometric traits, they may contribute to more than one. So we combine them together. We are hoping that will increase power to identify more genes that are associated with those traits. So, and the results confirmed this. So we were able to identify more, and especially this one study that was published 7 years ago. We were able to identify more SNPs that are associated with any of the traits, with either height or BMI or with the hip ratio or the adjusted for BMI. And this particular one is what we identified, and which this one is missed by the conventional GWAS study. If you analyze those traits separately, you won't be able to find this one. And if we combine them together, we were able to identify more. Make sense? Okay. All right. Now the next question is how to understand the mechanism that genetic variants influence pairs of traits. So we know that a lot of variants that are associated with more than one trait, and we can use the cross-phenotype association analysis to identify them. But once we identify them, we found those SNPs that are associated with more than one trait. We are wondering why this SNP is associated with more than one trait. What is the pathway? What is the underlying mechanism? So there are 3 types of pleiotropy can cause cross-phenotype association. The first one is what we call the mediated pleiotropy, one mediation. That means this SNP is associated with the first phenotype, and this phenotype associated with the second phenotype. Then if you do this GWAS separately, you will find this SNP associated with this phenotype and this phenotype. For example, BMI and type 1 diabetes, or type 2 diabetes. So if a SNP associated with BMI, and BMI is a risk factor to type 2 diabetes, so then this SNP is associated with type 2 diabetes as well, but the pathway is like this. Okay, And the second one is biological pleiotropy. That's an independent pathway. So this SNP is associated with two phenotypes independently. And last one is what we call co-localization. It's another type of biological pleiotropy, but at the gene level. So if we have two SNPs, one SNP associated with first phenotype and second SNP associated with second phenotype. But because those two, they are located near, so they are correlated. Then you would detect each of these SNP associated with both phenotypes, but that's another type of biological pleiotropy, but we call it co-localization. It's at the gene level. Okay. So how we can differentiate those different type of pleiotropy? So we use a method which is called the Mendelian randomization. And this is the approach to infer causality of an exposure for a complex disease outcome. This is a very powerful method to detect the causality, because most of the time, if we are looking at association, it's just association. You don't know the underlying mechanism. It could be the A cause B, B cause A, or it could be confounding variables. But this method allow us to detect the causality. It's because we start from gene, and gene does not change. So the SNP in your body, it does not change. If it has effect on the risk factor, and then the risk factor has effect on the phenotype, then we will be able to detect this pathway, because the confounding variable, it does not apply, it cannot affect your gene. Does this make sense? Because your gene in your body, it stays constant. So there won't be any confounding variable, because confounding variable your gene does not change. So we are able to detect what we are able to infer causality using this. Okay, so this is a study conducted by my student. We are looking at causal relationship between metabolic risk factor for breast cancer. So what we did is we look at 5 different metabolic risk factors. They are BMI, height, type 2 diabetes, HDL-C, and LDL-C. So HDL is high cholesterol level and LDL is low cholesterol level. Those 5 traits are metabolic risk factors, and we are looking at their causal relationship with breast cancer. So we use those risk factor associated SNPs to infer the causality for breast cancer. And we first performed the univariable analysis. So we were able to identify BMI and HDL-C. So BMI, the first one, and this one, HDL-C. Those two are associated, are causally associated with HDL-C. The rouge, this one?
C'est moi.
Peut-être on n'a plus d'internet.
Qu'est-ce qui se passe? Ok, no problem, go ahead.
D'accord. Comment je fais?
You have to share the screen.
Okay. Yeah, okay. Okay. I'm almost done. All right. The multiple okay. The multivariable Mendelian randomization analysis allow us to determine how the pathways through the 3 different trees. So after we put them together, then we found only HDLC has effect on BMI. That tells us that the pathway through those traits are because height and BMI are associated with HDLC, and HDLC is associated with breast cancer. Does this make sense? So height and BMI is associated with HDLC, and HDLC is associated with breast cancer. Yeah, so that's the pathway we found for those traits. So when we do univariable analysis, we found 3 traits that are associated with BMI. They are height, BMI, and HDLC that are associated with breast cancer. But we don't know how the association is. And when we put them together, do the multivariable analysis, we found that HDLC was responsible for the genetic association between BMI and height with breast cancer. That means BMI and height associated with breast cancer through HDLC. Does this make sense?
Which ones are responsible? HDLC.
HDLC, yeah. Because height and BMI are associated with HDLC, and HDLC is the one that is responsible for that is associated with breast cancer.
But that's only associated, not associated.
Well, causal association, because we started from genes, so there's no confounding variable. If we find the association, that will be the causal association. Make sense? Okay. I think this is my last slide. Okay. Let me summarize. Okay, so what we did is we first performed genome-wide association study. So it is a powerful tool to detect the association between genetic variants and traits in samples from population. And then we did a cross-phenotype association to identify that the SNPs that are associated with more than one trait. And lastly, we use a Mendelian randomization analysis to identify the pathways, the causal pathways, which is the mediation pleiotropy or mediated pleiotropy. And our results, so from the Mendelian randomization analysis, demonstrate that HDLC is responsible for the breast cancer risk. So it is causally associated with the risk of having breast cancer. Okay.
And this is new.
Yeah. This is the paper that I sent you last.
Yes, but I mean,
Yeah.
new about
We are revising and we are going to publish this with
no,
them.
I can imagine, but
Yeah.
that's very important.
Yeah. There's not very much math in it, more like application, and heavily rely on computational computing. Yes, because the SNPs, the genotypes are huge, and we have the sample size are huge. So usually we use I'm using a high performance computing system to do my analysis.